Hey guys, it's me again. Yesterday we looked at the steps involved in anaerobic respiration. We shall begin from where we ended yesterday when we looked at a brief about steps involved in aerobic respiration. So, yesterday we said that glycolysis or glycolysis is the first step in both anaerobic and aerobic respiration. And we say that this is an anaerobic step. It takes place in the cytoplasm and it is a 10-step process. The next one was the Krebs cycle, which takes place in the mitochondrion. And we said that the third step is electron transport chain. Oxidative phosphorylation becomes our fourth step. So we shall begin with the first step, which is glycolysis. So glycolysis, it takes its site is in the cytoplasm of the cell. In the cytoplasm of the cell, not of the mitochondria. So in the cytoplasm of the cell, it is an anaerobic process. This gives us reason why it takes place in what? In both anaerobic and aerobic processes of respiration. So it is also called the EMP pathway. EMP, the abbreviation EMP was named after the scientist, whereby E is Ibeden, M Moifel, Myhoff, then Pans, which is P. So we shall begin with the steps involved in glycolysis. So the first step, we see that a glucose molecule is converted into a glucose 6-phosphate. And now for this reaction, ATP is required, and an enzyme, phosphorylase, must be present. So as you can see that in this first step, this is an investment. What? This is an investment phase, because now we know that respiration is a process by which ATP is formed. But now we can see that from this first step that ATP is being what? Is being consumed. So we are now investing our ATP. We shall obtain it later. So the second step will be the conversion of this glucose 6-phosphate into fructose 6-phosphate. Now, these are isomers of each other. And that means for the conversion of this glucose 6-phosphate to fructose 6-phosphate, this is an isomer of glucose 6-phosphate, an enzyme isomerase is required. Isomerase enzyme is required. So, this will become the second step for glycolysis. Now, this fructose 6-phosphate, if we were to go to the third step, is converted into fructose 1,6-diphosphate. Now again, in this reaction, phosphorylase enzyme is required. And again, ATP is consumed here. ATP is consumed. That means ATP, which is adenosine triphosphate, will be converted into ADP, which is adenosine diphosphate. So now, if we are to go to the fourth step, we see that fructose 1,6-diphosphate will be broken down into fructose 1,6-diphosphate will be broken down into glyceraldehyde 3-phosphate, whereby these are 3 carbon compounds, and dihydroxyacetone phosphate. Now for the breakdown of this fructose 1,6-diphosphate, an enzyme aldosterase enzyme is required. Now when these two are formed, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, these two are isomers of each other. That means dihydroxyacetone phosphate can change into glyceraldehyde, and also glyceraldehyde can change into dihydroxyacetone phosphate, which are 3 carbon compound. Now this glyceraldehyde 3-phosphate will be converted into 1,3-diphosphoglycerate. Now in the conversion of this glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate, this glyceraldehyde undergoes an oxidation reaction by NADP, by NAD+. You can see it from the equation here. So it undergoes oxidation to form this 1,3-diphosphoglycerate. So we know that in any oxidation reaction, when one substance is being oxidized, the other is being reduced. That means that if this glyceraldehyde 3-phosphate is being oxidized to 1,3-diphosphoglycerate, NAD is being reduced to NADH2. Or you can also write it as NADH plus H2 plus H. So for more, so we shall continue with the 6th step from our next video. Don't forget to subscribe. Stay connected. Goodbye.